Well, first, thank you, Liam, for the invitation and for the nice introduction. This is the first manuscript we have with a single cell, and I think that will be not possible without the help of Liang and Xijing. And I will present the result we have. It's published in the Journal of Allergy and Clinical Immunology. And we are trying to characterize the allergic skin inflammation. It's on a mouse model. And I will present a little bit of introduction, what we are using this model and the disease I'm working on. Well, we're working with a mouse model of atopic dermatitis. This disease is very common. It's affecting around 20% of the babies and childs worldwide and in US as well. And recently, with the approval of new medication for this disease, more adults are coming to the clinics. And now it's around 10% of the adults are having this disease as well. This disease is characterized by red, itchy skin. Most of the parents complain about hearing the babies or the small child just scratching the whole night, which means that disturbs the quality of life of the parents as well and the patient as well. This scratching promotes a defective skin barrier because of the scratching and the inflammation induced by this scratching. And that allows the penetration of more allergens. And we think the skin is the main portal for the allergy. And this disease is characterized by type 2 immune response. That means you have increase of type 2 cytokines like IL-4 and IL-13. And you have recruitment of cells that produce these cytokines like T cells and mast cells and basophils, and as well the infiltration of eosinophils, which are hallmarks of this disease. We don't know exactly how this disease starts, but we believe that, again, as these patients are scratching a lot, that allows the production of epithelial cytokines or alarmins in the skin. And these alarmins will activate dendritic cells that capture the allergens that are introduced by the skin to induce immune response that is characterized by production of Th2 cells that produce all the type 2 cytokines like IL-4, 13, and 5. These cells are in circulation until you have a encounter with the same allergen, which their cells are recruited again to the skin and then attract more immune cells to produce all the features of the atopic dermatitis that I will show in the next slide. And these characteristics of ADs, first of all, you can see clearly in the epidermal hyperplasia, which is just the increase in the epidermis of the skin. If you compare with no lesional skin, it's small layers, but when you have atopic dermatitis, this is increase. This is as well characterized by spongiosis, which is this like edema in the epidermis. Now, what you can see, even though in the skin of no-lesional skin, you have infiltration of mononuclear cells, that is enhanced when you have this inflammation. And inside of these mononuclear cells, you have a lot of eosinophils, which induce eosinophilia in the tissue. For me, it's really interesting, this disease, because we believe that the atopic dermatitis is like the entry door for the allergies. And this is supported by epidemiological data showing that the atopic dermatitis eczema is very early in life. And then it's followed by food allergy, rhinitis, and asthma. And most of the patients that they have severe eczema or atopic dermatitis, they develop food allergy and rhinitis, again, suggesting that this disease is the entry door for allergies. And I think it's very important to understand the basic mechanisms here in order to prevent further allergic diseases. As I mentioned before, this disease occurs very early in life. And it's all the diagnosis is done by clinical criteria, which means we don't have access to many tissues to work for the basic research. For the reason the lab that I'm working on developed this model in mice, which try to mimic what's going on in the patients. We do tape stripping, which is just apply a tape and remove it, trying to mimic the scratching of the patients. And then we apply a patch with an allergen. Most of the time we use ovalbumin, which is a protein of the egg, and an allergen as well. But we can use any other environmental allergen like HDM or food allergen like peanuts. And these mice, they reproduce the inflammation observed in the patients. As you can see here, this is the mouse skin after epicutaneous sensitization. And you can see the epidermis. It's we have epidermal hyperplasia in those mice. We have infiltration of mononuclear cells and eosinophils, and you have the increase in the type 2 cytokines like IL-4 and IL-13. And interestingly, for us, this model reproduces as well what we see in patients. If you sensitize the mice through the skin and then you challenge them orally with the same allergen, these mice develop something like food anaphylaxis that we measure by dropping the body temperature of the mouse and the release of MCP1, suggesting that this model could help to understand a little bit the mechanisms behind the atopic dermatitis and the atopic march. When I joined the lab for my postdoctoral training, I tried to decrease the time we need to sensitize the mice, and we developed this short model of atopic dermatitis, and it's reproducing all the hallmarks that I previously mentioned. And I will show quickly some of the results we have. The mice we are sensitized with the allergen, they have increase in transepidermal water loss, which is a measurement of the barrier integrity, and it's similar to AD patients. Again, we have the epidermal thickness in the skin treated with the allergen. We have mononuclear cells, and we have increase in the immune infiltration. This is just showing here by flow cytometry, looking in the CD45 positive cells, that it's a pan marker for hematopoietic cells. And this is increased when you have the allergen. Even though you have damage in the skin, it's not reaching the same as if you apply the allergen, suggesting that the immune response is driven by the allergen. And I just want to you remember that you have very few immune cells in the skin compared with the no immune cells, that you will see why that's important later. And this infiltration of immune cells is characterized by increasing CD40 cells that the people believe is the major producers of IL-4 and IL-13 in the skin. You have eosinophils, that it's another hallmark of the disease. And again, you have the production of IL-4 and IL-13 in this treated skin with allergen, but not in the control, or it's decreasing the control. Using this model, we try to, again, understand the basic mechanisms of atopic dermatitis in order to get new biomarkers or possible targets to treat this disease or to prevent the disease. For this reason, we analyze the skin of the mice treated, as I previously mentioned, by single cell using the 10x technology. And I will show you what we obtained with this in this paper manuscript. First, with the help of Liang, we identify how many clusters we have in the skin of these mice in order to understand the disease. And as you can see here, we have we identify 47 different clusters in the skin that is treated with ovalbumin or control saline. And there are in both populations, there are, I mean, in both groups, all the different clusters are well represented. When we analyze the expression pattern of these different clusters, we identify 13 different cell types, including non-hematopoietic and hematopoietic cells. And as you can see here in the proportion of the cells, fibroblasts and keratinocytes are the major population that we can detect in the skin. And when we go for the immune compartment, it's very decreased, and it's fitting perfectly with what we know by flow cytometry. In this analysis, we identified these 13 different cell types that some of them are already published, but we encounter mast cell basophil population and neutrophils in our analysis that has never been described before in the skin. And we believe it's because of the inflammation induced by our model. We use, again, different just markers that we did manually just to see which cell population we include in this analysis. And one of the things we start to see when we compare our control group with the sensitized with OVA, we start to see an increase of the appearance of these two populations. One is mast cells basophil, that it's maybe one cell in the Salin group, and the other population is neutrophils. And again, that we believe is because of the inflammation induced by the allergen. You are attracting these cells. But apart, if it's not evident in this analysis, we just look for the proportion of the cells we can identify with the markers we select. And we can see that the sensitization with the allergen increase the number of T cells, macrophages, and dendritic cells, fibroblasts, periocytes, mast cells, and neutrophils. Again, this is fitting with what we know in our model, that you have infiltration of the immune cells, and that's where you have increase in the dermal portion of the skin, which is mainly consistent in fibroblasts. And that's where we know we have this angiogenesis that is induced by the periocytes. As we know, we have this difference in the number of cells or proportion of the cells infiltrating the skin or resident in the skin. We were interested to characterize better all the population we can identify. For this reason, we start with the major population in the skin that we can detect in our analysis, and those are the fibroblasts. In this analysis, we identified 16 different states or types of fibroblasts containing well-known collagen 1, 8, and 9 fibroblasts, the dermal papilla, myofibroblasts, and lipofibroblasts. And when we look for the fraction of the cells, we don't see big difference between the control and the mice sensitized with the allergen, suggesting that the proportion of the cells is similar in this model with or without the allergen. However, when we start to look for the differential gene expression, we can see that some of these population of fibroblasts start to express chemokines for immune cell like CCL8, TCL2, and CCL7 that is well known to attract CD4 T cells. We have as well CCL5 that is well known to attract neutrophils. And we start to see as well the production of collagens and osteochondrin, which is important for the skin, of the production of the skin. And interestingly for us, we start to see in these fibroblasts treated with OVA that they express, so they increase the expression of IL-33, that it's believed that it's only expressed in epithelial cells, but we can detect in our analysis, suggesting that the fibroblasts play an important role in skin remodeling and as well the attraction of the immune cells. Oh, sorry. The second population we characterize is the keratinocytes. And again, looking in different manuscripts, we have all the layers that's supposed to be in the skin. We have a basal layer, a spinous layer, granular layer, and all the components of the hair follicle. We have as well sebaceous gland. And again, we see a little bit of decrease in the fraction of the cells in the basal layer and the spinosal layer, but it's not that dramatic. And I don't think it's significant difference between the two groups. Again, it's well known that in atopic dermatitis, they have this problem with the keratinocyte differentiation and final differentiation that induce this skin barrier defect. Consistent with that, our analysis identify decrease in a lot of keratins in different layers of the skin. As you can see here, keratin 10 and keratin 2 is almost present in all the layers of the skin. And as well as the final differentiation of keratinocytes like filaggrin, loricrin, and transglutamin 3, it's decreasing in mice easy sensitized with OVA, suggesting that the keratinocytes in this model are following similar pattern than the patients with atopic dermatitis. Finally, we went to the endothelial cells, and we can identify two kinds of endothelial cells, lymphatic and vascular. Again, we don't see big difference in the proportion of these cells. However, when we start to look for the gene expression profile, we identify selectin P and CXCL2 that are increasing the allergen sensitized mice, suggesting that these endothelial cells are important for the recruitment of the immune cells. Okay. With this, we just characterize the no-immune compartment of the skin, and with our analysis identify that fibroblasts are important for the chemokine production of the chemokines that are important for the attraction of the immune cells, and as well they are important for the skin or the tissue remodeling observed in the skin of AD patients, and as well the endothelial cells that are important for the attraction. And it confirming that keratinocytes in our model are fitting with what as is described in the atopic dermatitis patient. They have dysregulated the expression of keratin and decreased expression of genes that are important for the barrier integrity. As we are interested in immunology of the atopic dermatitis, we look in the immune cells. And as I mentioned before, you have increase in this population of the immune cells in the skin of OVA-sensitized mice, like T cells, macrophages, dendritic cells, mast cells, basophils, they appear in the skin the same as neutrophils. And again, this is consistent with increase of immune infiltration we have in the skin of these mice. When we look to the different clusters, we observe that in the T cell cluster, we have increase in the transcript for CD4 and decrease in the transcript for the genes that codify for the receptor D or the TCR delta in T cells. And we confirm that by flow cytometry. Here we can see that the number of, or the proportion of CD4 T cells infiltrating the skin of these mice is increased compared with the control group. And the proportion of gamma delta T cells, or cells expressing the receptor delta, that are decreased when you treat the mice with the allergen. We observe as well the analysis that our T cells are expressing more HR, that is a transcription factor that is really, really important for the production of IL-22. And we confirm that in our model, we have increase in the production of IL-22 CD4 T cells or Th22, that it's always also in the skin and lesional skin of patients with atopic dermatitis. The other thing we saw is the IL-13. It's increased in the T cells of these of the mice treated with the allergen, and it's confirmed using reported mice for IL-13 that express GFP when IL-13 is produced. As you can see here, when we have sensitization with the allergen, the proportion of IL-13 producing cell is increased, confirming the result we observed by single cell. And the other thing we didn't detect increase in FOXP3, which is the hallmark of regulatory T cells. However, we have a signature that increase the expression of CTLL4 and DR3, which are expressed by regulatory T cells. And we confirm that the regulatory T cells are increased in our model using reporter mice for FOXP3 that produce YFP or express YFP when these cells express FOXP3. But you can see here we have increase in the number of cells that express FOXP3, indicating that the epicutaneous sensitization with allergen induce an increase in regulatory T cells. And as well, as I mentioned before, we have this increase in the basophil mast cell population that we couldn't see in the saline group in our analysis. But when we analyze by flow cytometry, we can see that they have decreased numbers of basophils that are characterized by the expression of or the fixation of IgE in the surface, which is here, and lack of this marker, CD117, that it's a c-KIT. There are increased when you treat the mice with OVA, and as well the population of mast cells that express both markers, indicating that the epicutaneous sensitization increased both population mast cells and basophils compared with the control group. When we analyze the expression of these cells, we notice that IL-13 and IL-4 are expressed in those cells. That it's known that they express the cells, but we didn't know that those cells will be increasing the expression with the epicutaneous sensitization. Having all these results and interested in this IL-4 and IL-13, that there are many of the pathological features of the AD are consequence of the action of these cytokines, we look which cells they express more IL-4 or IL-13. Now you can see here when we compare the population mast cell basophils against T cells, the basophils of mast cell population is the only one expressing IL-4 and is expressing higher levels of IL-13 compared with T cells. This is something for us, it's a little bit surprising because all the people think that IL-4 and IL-13 it's mainly produced by T cells in this disease. What we observe in our analysis is we identify mast cells, basophils, and neutrophil population in the skin that again it's not has not been reported before in the skin, and we believe it's because of the inflammation induced by this allergen. We have increase in gene associated with Th2 polarization like IL-13 and Tregs like CTLA4, and we confirm that by using reporter mice and suggest that epicutaneous sensitization or inflammation in the skin induce the increase of this Th2 population and regulatory T cells. And as well, the more interesting results we get is the mast cells and basophils rather than the T cells are the major producer of these cytokines, IL-4 and IL-13 in the skin. Again, this is a mouse model. We don't know that we can export or translate these results in humans, but it's really interesting to look at these populations in the actual human disease. One of the reviewers asked us if, I mean, having these concerns about how your model really reproduces the human disease, if we can compare, there are 3 different papers in atopic dermatitis patients looking by single cell. And I just show you here a couple of examples using the event that it's developed by Liang. And we have some population like the fibroblasts that are, we believe they are very similar between our study and our model and the human AD. As you can see, there are some genes that are regulated in the same way. But when we look in another cluster like keratinocytes, we have some discrepancies between our study and the human studies by single cell. But we identify as well that those studies, some of them, there are contradictory between them. And I think it's by maybe multiple reasons. One, it's how they process the tissue just to get the single cell suspension, and as well the way they do single-cell RNA-seq barcoding and subsequent analysis is different. And I think it's one of the main issues of this technology, that there are no kind of established pipelines that all the people is using. Is everyone is doing in different ways, the preparation of the cells and the analysis as well. The other thing we analyze, it's using data sets from microarray or RNA-seq. We identify some of the genes that are upregulated or downregulated in atopic dermatitis patients, and we compare with our data sets. And we identify some of the genes that are upregulated in AD that are fitting with some of the genes or orthologous genes in mice. And we can identify where this gene is expressed in our data set. And I think in conclusion for this study, we can say that we can apply this technology in our model in order to identify new molecular mechanisms leading to the disease, and that will to try to identify novel therapeutic targets in AD. However, I think we have, I need to mention that we have a lot of limitation in our study. One, it's, we didn't identify eosinophils, that it's one of the cells that predominantly are in the skin of the patients. And as well, we didn't identify ILC2s, so type 2 innate lymphoid cells, that people think play an important role in the allergic skin inflammation in mice and humans. And we didn't see any transcript for IL-17 and IL-22 that we know they are there by our previous studies, and there are in atopic dermatitis patients as well. And I think one of the main issues we have is the limited number of immune cells that we can analyze. And I think that's the main reason why we don't have more granular analysis, that we can identify type 2 cells, Tregs, or Th2, Th27 cells. And as we'll see how they are different if you are in presence or absence of the allergen. And the other thing is we couldn't split the populations between regulatory T cells and conventional T cells, for example, or gamma delta T cells that are all together in one cluster. And as well, the mast cell and basophils, we don't know which cells are expressing which transcript. If it's mast cells mostly expressing IL-13 and basophils expressing IL-4, we don't know. And I think it's important to know to get more targeted therapeutics for this. And the other limitation of this study is we have just a single time point, which means it's just when the disease is established. I think it will be interesting to look at different time points to see the kinetic of the immune response and how the structural cells respond to this inflammation as well. To try to solve one of these issues, I start to do some analysis with sorted cells. I first start with CD445 positive cells, which are here, all this population. As you can see, we can see better the distribution of the immune cells in the skin. And as well, you start to see more granularity in the analysis. When we look for the lymphocytes or T cells, here now we can detect where are the conventional T cells and regulatory T cells. We identify ILC2s and ILC1s as well, that we couldn't identify in our previous analysis when we use the whole skin. And I guess it's important to know the proportion of cells you are trying to analyze when you are using this single cell technology. I just want to say thank you to my mentor, Raif Jeha, for all the support and all the help with this project, all the people working in my lab. Of course, Liang and Xingji to help me with all the analysis. Aviv for helping us with the first barcoding of the samples, and all the funding agencies that are supporting my research and research in Dr. Jeha's lab. And all of you for attention, and I will be happy to answer any question.
Great, thank you, Manuel. It's really great talk. So please just either say raise your hand, I will try to unmute you, or you can type your question in the Q&A session. I think I'm going to give people maybe 1 minute to think about a question. So let me ask one simple question, Manuel. So just wondering, for this project, did you think about using the result from the pathway analysis and the goal enrichment analysis? Is there any reason that, say, for example, because the limited of sequence steps, I think that's kind of common problem for single cell RNA-seq. We cannot have a very high sequence steps compared with the bulk RNA-seq, right? So is that the reason that we don't have a lot of differential expressed genes, and so that's why it's not possible or it's not good to do the pathway or goal enrichment analysis?
Yeah, I think one of the main issues is the power we have for the statistical test. Again, we are very, very limited by the number of cells we have, at least for the immune compartment, and I think it's not, you have very few genes that they are differentially expressed and significant. It's very hard to get, hits in a pathway analysis. And I think it's one of the main issue that we are trying to solve. You can sort a specific cell type and say, okay, we can focus in the cells we are interested in order to get more deep analysis, as you say, or more granular analysis.
Great. One more question is that do you have any plan, basically any future plans for this project, maybe some new technologies or for example, spatial transcriptomics analysis, for this project, or any other plans you have? What you did, right? You actually focus on the dendritic cells, and then also I think there's another single cell RNA-seq data from the dendritic cell, I think. So do you have any further plans for after this project?
Yeah, again, we have a very powerful technology, the single cell. And I think we have the opportunity, my own interest is just to know how the cells are communicating in the skin. Because most of the people think it's just one cell doing all the job. And I think it's not correct. And I think that you can, using this technology, you can really, start to ask questions how the cells are communicating in the skin in order to prevent inflammation, like regulatory T cells. I don't know if they are affected in this model, or how other cells like basophils and mast cells are producing IL-4. I didn't present anything, but I know that T cells are, even though they are not the major source for IL-4 and IL-13, are really important for the induction of these cytokines by mast cells and basophils, which, that gives you a network of interaction in the skin that could lead to a new potential biomarkers, and as well molecules that you can use as possible therapeutic targets.
Got it. Great, thank you. Any other question from the audience? Please raise your hand. Okay, we have one question from Jose. Let me just unmute you. Jose, I think you can talk now.
Can you hear me?
Yes.
Excellent. Manuel, thanks for sharing that with us. I wanted to kind of tie together the reply that you just gave, Manuel, with what Liang raised before in terms of, how you can kind of go deeper into the single cell data that you've generated here. And I think the point you just raised is really important, right? Which is you're interested in the interactions of these cells. And a lot of the tools that exist for bulk transcriptomics or were designed with bulk transcriptomics in mind can sometimes fall short, right? And there's these shortcuts essentially that try to get you towards interactions from single cell data, but they're still trying to impose that prior knowledge. So I think that there's a real need in the field to be able to actually learn from the single cell data, right? What these interactions might be, because as you suggested, right, you might have certain ideas or certain experiments that have suggested already, okay, here's a key node, right? But how do we work those priors into our single cell analysis, and then how do we extend them, right, to find other things like them that we may not be aware of just yet? So curious to hear thoughts from either of you in terms of what you might be what that might spark, I guess, in terms of discussion.
Yeah, I mean, it's again, it's very good point, Jose, you are making, and I think I am a biologist by training.
Same, yeah.
and I think with all the amount of data we're getting with single cell, we need someone that kind of what's an intermediate state to say, okay, yes, I do the computational work, but I understand the biology behind. And I think it's one of the, I guess, one of the main issues in the field, that we are very good computational people doing all they can do to do the tools. But I think there are no good communication between biologists and technologists to say, okay, we can join forces with the knowledge we know as biologists.
Yeah.
To, feedback what you're trying to.
Yeah. I think there's a really interesting opportunity there that you're commenting on. And, I think that the other point to raise is that I think even purely from the computational side, there's a lot of room for innovation, actually, in terms of the way that we analyze single cell data currently. And, it would be great to discuss maybe offline in terms of, okay, how do we learn some of the, interactions that you're suggesting, right, might be important without needing to go back to kind of known pathways that we understand, right, and imposing that on our data. So I think that there's some interesting computational opportunities, but the point that you raised, right, of kind of these individuals with really hybrid capacity, right, of moving back and forth, getting the domain expertise for the computational biological tools, completely agree with you. Very, very important to really maximize these data sets. And last point I'll make is I think in terms of the sequencing depth versus cell number, I mean, I think that if you were to probably, and here of course there's certain realistic considerations of cost, et cetera, in these experiments, but if you were to acquire 20,000 T cells, then probably a lot of these subsets and a lot of these innate lymphoid cells that may be 3 or 4 cells in there, they start to fall out of the data, right? Because the way that we analyze these data in terms of clustering, it's still relativistic, right? You need to basically say, okay, I have enough cells here to make a group, right? So you probably have some Tregs, some ILC2s, et cetera, that are hiding there. It's just not enough to really make that separate out into its own group. So, the way that we can build maybe atlases in more unified fashion, right, which you suggested when you started to discuss the human versus mouse and all of this, I think will be really important to hit those, critical levels of cell numbers and say, oh yeah, obviously, like here's my Tregs, and here's my ILCs, right? No question. And, maybe here's another cell type that I didn't know to look for. So, I think it's a very exciting study that you've done, and I think a lot to build on there, either with time or additional cell number. So thanks again for sharing.
No, thank you, Jose, for your comments.
Great. So Manu and Jose, I'm not sure whether you heard about the cell phone database. So that's basically its database. Okay, you did hear about that.
Yeah,
maybe we can look at that.
I'm nodding here too, but I only see my own sticker staring back at me. So yes, I am nodding as well. This is actually what I was referring to, though, where those receptor ligand databases, right, they're imposing prior knowledge on the system. And actually a lot of the prior knowledge that comes in is suboptimal, you know, because it hasn't been curated sufficiently. Either there's interactions that are there that maybe aren't true, or there's interactions that are missing, right? So I think that this is actually one of these kind of gaps right now where I think curation is a must of what those prior interactions are. And then secondly, you know, how can we actually leverage more of the sheer number of single cell studies that are coming out to learn, you know, potential interactions without having to impose that knowledge?
Yeah, but because I think, I mean, again, as biologists, you know, you
Yeah.
have a threshold as well, not
Yes.
because they are expressing one receptor and the other one in the ligand.
Absolutely.
That they interact, and I think that's missing as well. That you are
Yeah.
not
Yeah.
really saying, okay, we put thresholds that maybe
Yep.
increase the usefulness of this analysis.
Yeah. And at the end of the day, you know, the point that Long brought up in terms of the spatial transcriptomics, I think that's going to be really important as well, right? Because obviously, if you can start to integrate across both of those data modalities, or not even spatial transcriptomics, but actually, you know, with a 5 or 6 IF panel, you can start to get pretty far in terms of saying, okay, you know, are my predictions correct? Or can I now, based on this imaging data, actually find this in my single cell data, right? And so forth.
Yeah, exactly. And it's spatial and
yeah.
yeah.
Yeah.